Hi everybody, we're going to finish up chapter 7. So we left off on two classifications of radiation interactions on a cell, direct and indirect. Because the human body is 80% water and less than 1% DNA, all of the effects, essentially all the effects of low LET irradiation in a living cell result from indirect action. And your book states approximately 2/3 of all radiation-induced damage is believed to be ultimately caused by the hydroxyl free radical, which is a byproduct of this indirect action. So this is the most damaging one. Okay, so this is just a picture of, to illustrate direct versus indirect. Direct action, the X-ray photon can hit the DNA helix or any macromolecule, RNA, DNA, proteins, enzymes, and have a direct interaction with those elements, with those atoms. Indirect, it's going to hit water and react with the water by producing ions and free radicals, and those are going to have reactions with things that they encounter, like DNA, RNA, proteins, enzymes. But since it's not the photon hitting those macromolecules, and it's one of the byproducts of this ionization of water, it is called indirect. Okay, so the radiolysis of water, which is this action from the photons interacting with water, and since we're mainly water, this happens a lot with ionization. So you're going to have the water molecules. They're going to produce free radicals or ions as a result of the production of free radicals, there's undesirable chemical reactions. Biological damage can occur. Okay. And there's also toxic substances that can form from these interactions. This slide is an overview of the indirect action on water. So it shows the photon coming in and hitting the water molecule, which can produce ions and free radicals. The ions it produces are a hydrogen ion and a hydroxyl ion. The free radicals that it can produce are hydroperoxyl and hydroxyl and hydrogen radical. So of the substances, the free radicals are the most dangerous. And what can occur with the free radicals is that hydroxyl can combine with another hydroxyl, and that can form hydrogen peroxide. And that's a toxic substance to the cell. And again, remember, approximately 2/3 of all radiation-induced damage is believed to be ultimately caused by this hydroxyl-free radical. So that's important, remember that. So here we have hydrogen peroxide that can be formed, which is very poisonous, and we have these free radicals bouncing around in there that can do this indirect damage to these macromolecules, specifically DNA, RNA, protein, enzymes. The hydroperoxyl radical and the hydrogen peroxide are believed to be among the primary substances that produce biologic damage directly after the interaction of radiation with water. So something to remember, essentially all the effects of low LET irradiation in living cells result from indirect action. Okay, so some of the effects of ionizing radiation on DNA. You can have a single strand break where it comes in and transfers energy and ruptures these chemical bonds. This is said to be repairable. It can be repaired by our repair enzymes that we have inside our cells. It's called a point lesion when you have one break like this. Okay, and this can be repaired. So this next slide shows a double strand break. So say there's further exposure of the same macromolecule by that ionizing radiation or that indirect radiation, and it can lead to additional breaks in the chain. They are farther apart, but they're still threatening the life of the cell. Repair can take place, although not as easily, and this is what that looks like. So it's just the same kind of breakdown of the molecular bond, but in a space somewhere else along the DNA chain. So we have types of DNA damage are going to be the single strand break, the double strand break, and this third type is the double strand break in the same rung of DNA. This is what that looks like. Okay, so if this results, each new portion contains an unequal amount of genetic material. And if this were to divide, each new daughter cell will have the incorrect amount of genetic material. This results in cell death or the impaired functioning of the new daughter cell. And that's all we're going to cover on the DNA damage. Okay, so the next thing we're going to talk about is target theory. Okay, so when they talk about target theory, they are talking about the concept of every cell has cellular components inside of it, molecular components. And there's usually more than one of each type. And if one gets damaged by radiation or being irradiated, then another one can step in and support the function. However, some molecules inside cells are unique. There's only one of them. And if that were to get damaged, then the cell is compromised. And this is called a key or a master. It's the concept that there's a key sensitive molecule, and for the cell to die after radiation exposure would mean that its target or key molecule must have been inactivated or damaged beyond repair. So in target theory, the target is considered to be an area of a cell occupied by a target molecule, usually DNA. So target theory holds that the cell will die after exposure to ionizing radiation only if the master or key molecule is inactivated in the process. Experimental data strongly support this concept and indicate that DNA is the irreplaceable master or key molecule that serves as the preeminent vital target. Okay, that's all you need to know about target theory. So this next slide shows effects of irradiation on the entire cell. Of course, radiation can affect the cell adversely, and the damage to the cell's nucleus will reveal itself in one of those following ways. Okay, next we're going to get into the cell survival curves. So cells vary in their radiosensitivity. A classic method of displaying the sensitivity of a particular type of cell to radiation is the cell survival curve. This figure shows two curves. One is for high LET and one is for low LET. So the curve for low LET radiation shows very little change in survival at low doses. This just indicates that the cell is able to find and repair some of that damage. So that's called a shoulder. Okay, you can see where the shoulder is on that low LET curve. At higher doses, however, the repair mechanism is overwhelmed. And for the high LET, no survival shoulder exists. Okay, if damage occurs, it is usually so extensive that it is irreparable. So this next slide is cell radiosensitivity. Remember, cells vary in their degree of radiosensitivity. Immature cells, which are non-specialized, they're undifferentiated, they undergo rapid cell division, are very radiosensitive, versus more mature cells, which are specialized. They're highly differentiated, they divide at a slower rate, they are less radiosensitive. Okay, or insensitive, they can be called. Okay, some examples of the radiosensitive cells: basal cells in the skin or epithelial cells, blood cells such as lymphocytes and erythrocytes. Lymphocytes are the most sensitive, radiosensitive cell in the human body. Also reproductive or germ cells, okay? The radio-insensitive cells, brain cells, muscle cells, nerve cells. This is another slide that gives you a little more examples. Most sensitive lymphocytes, gonads. Keep in mind, lymphocytes are the most sensitive, radiosensitive blood cell in the body. Okay. Intermediate are your intestines, your the cells that line your intestines, your skin, your epithelial cells, the lens of your eye, your thyroid, and then the least sensitive cells: muscle, nerves, spinal cells, brain cells, which are in the central nervous system. Okay, don't forget the oxygen enhancement effects on cells. So oxygen enhances the effects of ionizing radiation by making the tissue more radiosensitive. So oxygenated tissue has more radiosensitivity than hypoxic tissue. This can cause problems in radiation therapy. For instance, when a tumor has a low oxygen level in the tumor, the radiation that they are using, the therapy to try to kill those cancer cells, it will not work as well. So what they do is they use high pressure, hypobaric chamber or an oxygen therapy to force oxygen into the tumor cells, thus raising the oxygen level, and now they are more radiosensitive to the radiation therapy. So just a little side note, it increases tumor radiosensitivity. Okay, this is about the law of B and T. I think it's Bourgonais and Tribando. This law states that the most pronounced radiation effects occur in cells with the least maturity and specialization or differentiation, the greatest reproductive activity, and the longest mitotic phases. So they made this observation by studying germ cells of rabbits that had been exposed to X-ray. They established that the radiosensitivity was a function of the metabolic state of the cell. And even though this was originally thought to be just on germ cells, it has now been proven that it is actually true for all types of cells in the human body. And so just remember, least maturity and specialization or differentiation, greatest productive activity, longest mitotic phases. Okay. Other cell types of radiosensitivity are going to be the lymphocytes. They're the most radiosensitive blood cells. When they get damaged, body loses its natural ability to combat infection, becomes more susceptible to bacterial and viral antigens. Also platelets, you lose your ability for clotting. Okay, this is more types of cells that are radiosensitive, epithelial tissue, and that is because they are constantly regenerating, makes them very radiosensitivity. Opposite, muscle tissue. It's relatively insensitive to radiation, so it's not, they are mature, so it's not immature, it is not constantly dividing, it's specialized, okay? Also, nerve cells are highly specialized, so they are radio insensitive. On an off note, fetal developing nerve cells, because they're developing, they're more radiosensitive than the mature nerve cells of the adult. So studies indicate 8th to the 15th week of gestation in the fetus is the maximum radiosensitive time. And that's because everything is happening with the nervous system. Okay, so, and reproductive cells, of course, very sensitive. And that's all we have for Chapter 7. Just remember some of the highlights of the reading. You just need to know about your RBE, which is your relative biological effectiveness, that it is using the radiation weighting factor. Remember what oxygenated tissue does. It makes it more radiosensitive when it's irradiated. Know the difference between direct and indirect types of interactions from radiation. You don't need to know the specifics about the DNA types and the chromatid types of malfunctions, but you do need to know target theory, okay? And you do need to know that curved linear response, okay? Thank you.